Understanding Microscopes by Robert, Holly, and Kai Chi. The goal of this lab is to design and build two different microscopes. The first microscope must produce a real image on a screen. The second microscope will produce an image that must be viewed through an eyepiece lens. Using both of these designs, the goal is to figure out if attaching a CCD camera to an eyepiece of an old microscope will produce an image. The materials we used for this lab were a convex lens with a 75mm focal length, a convex lens with a 150mm focal length, a light source, a viewing screen, and an object, which was a crosshair with an arrow.
For our first setup, we used one objective lens and one eyepiece lens, and they were lined up horizontally. Both of them are concave lenses. As you can see on the picture, the light source was placed behind the object, and the viewing screen was placed behind the eyepiece lens. Our object, which is green on the picture, was placed within the focal length of the objective lens. And this setup gave us a real image produced on the viewing screen and a virtual image produced behind the object. Our second setup was very similar to the first setup. One objective lens and one eyepiece lens were lined up horizontally, and both of them were concave lenses. The light source was also placed behind the object, but the difference this time was the image was viewed through eyepiece lens, and the object was placed outside of the focal length of the objective lens. As an outcome, this setup gave us a virtual image produced behind the object and a real image produced between the two lenses, which is why the real image could not be formed on a viewing screen.
We used two equations during this lab. We used an equation on focal length and an equation on magnification. The equation on focal length involved the distance from the image to the lens and the distance from the object to the lens, as well as the focal length, while the magnification equation involved magnification, the distance from the object to the lens, the distance from the image to the lens, as well as the distance of the height of the object originally and the height of the image. The key difference between the two designs of the microscopes we built or how you viewed the image and what kind of images they created. With our first design, you viewed the image through the lens and it created a virtual image that was placed outside the focal length. With our second design, you viewed the image on the screen, which created a real image and that was placed within the focal length. When working with one lens, the orientation of the real image is inverted, which is how the old camera works. But with two lenses, it allows us to create a correctly oriented real image. Also, the objective lens and the eyepiece lens enables us to focus the image and zoom in, making it magnified. It helps to enclose the light path from the objective lens to the eyepiece with a solid box, because it blocks out a lot of the natural light coming in from the surroundings, which allows the lights to appear more intense and appear much more clear to the person looking at them. And when looking at a sample with a microscope, our eyes are viewing a virtual image. This is because the object is placed within the focal length of the objective lens, which is evidence that the image perceived by our eyes is in the same direction. But obviously it thus is a virtual image, and it also is enlarged because of this. In order to project the microscope's image onto the detector of the CCD camera, we would use Design 1. Design 1 produces a virtual image, so that is why we could attach the camera lens to the microscope, since it would look right through the lens, just as our eye did. For the microscopes of the hypothetical lab, we would not need to make very many modifications. The only thing would be was once we have the camera attached, maybe adjusting how far the object is from the DI or DO of the object, just to make sure it's in focus and the image is clear. And because of this, we would not need to purchase new microscopes for the lab.